Okay, let's talk more about osmosis. Osmosis is the diffusion of water across a selectively permeable membrane. But how do we talk about that? Well, we use terms like hypotonic and hypertonic and isotonic. Here, the prefix hypo is referring to a solution with a lower solute concentration than water. The prefix hyper is referring to a solution with a higher solute concentration than water, and isotonic is referring to solutions with equal solute and water concentration. When we use these terms, we're talking about usually two solutions and comparing them. For example, a solution with more solute than another is hypertonic. A solution with less solute than another is hypotonic. So we usually compare two solutions to use these terms. Let's look at the example on the left, a U-shaped test tube with a membrane in the middle that is impermeable to the solute. So those purple dots are not going to move, but the water can. And the water will move from the left to the right, or from where there is high concentration of water to where there is a lower concentration of water. The reason that occurs is because water moves towards the area where there is more solute or less concentration of water, because during osmosis it passively moves and it's moving down its concentration gradient. So let's look at some more examples. Let's look at these two examples at the bottom here. We want to know, does solute move through the membrane and does water move through the membrane? And then we want to know, if they do, which direction. For the example on the left, we have a semi-permeable membrane that is impermeable to solute, so the solute doesn't move. But the situation on the right has a membrane that's permeable to solute and water, so the solute does move. And for both membranes, both situations on the left and right, they're both permeable to water, so water does move. But what about directions? Well, for solute movement, it's still passive. We're not adding energy, so we're still going to be getting movement from high to low concentration. And for both situations, since they mirror each other, except for their membrane permeability, we're going to see that the solute, where it can move, on the right example, moves from the right side, where there's more solute, to the left. And in both situations, since water can move through both membranes, it's going to move from the left, where there's less solute, to the right, where there's more solute. We can also compare the situations up at the top, labeled A, B, C, and D. Solution A and solution C have a different amount of dots, which represents the solute. Solution A has more solute than solution C, so it is hypertonic to C. Solution C compared to B is hypotonic. Solution B compared to D is hypertonic. And solution D to A is isotonic. So those are ways to use terms hypotonic, hypertonic, and isotonic to talk about two different solutions and how much water they have in comparison to each other. But what does that mean for cells? What does osmosis mean when water rushes into a cell or it leaves? Well, it has to do with causing a cell to shrink or swell, and that has to do with tonicity. So with an isotonic solution, we really see water move in and out of a cell in an equal amount, and it shouldn't change the shape of a cell. But if you put a cell in a hypertonic solution, and there was more solute outside the cell than inside, all the water inside the cell would rush out. That would make the cell shrivel or crenate, and that can cause a cell to lose its function. With a hypotonic solution, a cell placed in this kind of surrounding of just water would take in a lot of water. That water would move from outside to inside where there is less water, and that cell would swell. Cells that are animal cells don't have a protective cell wall, so they would swell to the point where they would eventually bust open, and that's called lysis. So it's important to remember these terms because it plays a role in the functionality and the preservation of cells. Intravenous solutions of different tonicities can be given to patients suffering different ailments. Usually we administer isotonic solutions. They're the most commonly given because it just helps us maintain blood volume, and it helps especially to increase blood volume quickly, but it shouldn't affect the cells. Hypertonic solutions, though, could be given to swollen patients that really need to pull water back into the blood. so we need it to leave the tissues and move into blood. Hypotonic solutions really should never be given. They can result in dangerous lysing of red and white blood cells. There's not many situations where you would use them. So here we're looking at a summary of all these processes that we've paid attention to thus far. Things like simple and facilitated diffusion and osmosis. They all use the energy of atoms, or kinetic energy, but none of them use ATP. None of them use cellular currency. So it's just the movement of molecules, and molecules are always moving. For simple diffusion, it's where we see something go through the plasma membrane, in or out, without the use of a channel or carrier protein. For facilitated diffusion, we're going to need that channel or carrier protein, and it's more for moving things like ions or something like glucose or amino acids. And with osmosis, we're not moving solute like we are with the diffusions. Instead, we're moving water. And whether it's a solute or water, we're moving water from high to low concentration or solute from high to low concentration. Water can move straight through the membrane, but it usually moves through something called an aquaporin. Aquaporins are just special channel proteins for water. So that's our basics for passive processes. We don't want to ignore, though, active transport. Active transport moves things from low to high concentrations, or against their gradient. This requires cellular energy. It requires ATP. This is to move really large amounts of things during a process like endo or exocytosis. So bringing stuff in vesicles or getting rid of it like a waste product. But anything that's too large for a channel protein, or can't just dissolve through the lipid bilayer, or can't move down its concentration gradient, might need to use active transport. Different types of transport proteins, oftentimes called transport pumps, may be utilized and are powered by ATP in order to achieve active transport. And that's what we're going to be looking at in the next video.